Hello, and a very warm welcome to today's Nucleic Acid Insights webinar titled Addressing the Challenge of Screening DNA-LNP Formulations for Targeting the Brain. I'm Casey Nevins, an editor at BioInsights, and joining me today are Ben Knappett and Razieh Karamzadeh. They will present on how using Unchained Labs' Sunscreen and Sunshine systems simplifies the rapid development, screening, and scale-up of DNA-LNP formulations. This enhances the efficiency of transitioning from in vitro screening to in vivo studies and GMP production. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions to Ben and Razieh using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. Now I'd like to introduce our presenters. Ben, with a background of nanoparticle synthesis, is the product manager for LNP solutions at Unchained Labs. There, he focuses on automated, high-throughput solutions for drug delivery, vaccines, and gene therapy. Razieh has a background in molecular cell biology and biophysics. Her postdoc research is on developing high-throughput screening for DNA lipid nanoparticles using automated microfluidic systems and advanced culture models for brain-targeted LNP screening. So without further ado, I will hand you over to Ben to kick us off with the first presentation.
Thank you for that introduction. I'm really excited to be participating in today's webinar, which is going to take a look at addressing the challenges of formulation screening when it comes to targeting particular tissue types, in this case, glioblastomas in the brain. But before I hand over to Razieh, I'm going to spend a couple of minutes on the challenge at hand when it comes to developing nucleic acid-based therapeutics. So when researchers are developing LNP-based drug formulations, typically we're looking to start by developing a functional particle formulation to deliver the nucleic acid cargo, and then to optimize a preclinical process for producing larger volumes of these particles. Once clinical trials begin, the LNPs are going to be injected into populations of humans and increasing numbers of humans as we move through towards the commercial release of the drug, ultimately aiming for regulatory approval and commercial manufacturing. So this requires an increase in production volume and a regulated particle production in a GMP environment. So each of these different development stages taking place has a different set of requirements, might well be taking place in different labs, and ultimately needs to be able to transfer reasonably seamlessly between those different development steps in order to make the development process as efficient as possible. So the Sunny Suite from Unchained Labs offers a unified solution to the particle production throughout each of these stages, addressing the particular needs of each development stage. So Sunscreen enables users to screen up to 96 low-volume formulations in a single automated run, accelerating the discovery of lead candidate formulations for further development. Sunshine then takes the formulation identified from Sunscreen and enables the rapid optimization of the particle production process, with a view towards then also carrying out continuous production for larger preclinical production runs on the same instrument. Finally, Sunbather then takes the wheel as you enter a GMP manufacturing space, producing the same high-quality particles with that nailed-down formulation and process, but now meeting all the requirements of a GMP environment. So these instruments all share a consistent pumping methodology and range of microfluidic mixer types called Sunnies, which means that it really is easy to transfer from any one of these instruments to the next, facilitating the transfer between different laboratories, different manufacturing suites as well. Today, though, we're going to focus on step one of this workflow, which is screening. So the potential scope for LNP formulation screening is truly enormous. There are some variables that are more commonly considered, such as the nucleic acid cargo itself, as well as the four different kind of key lipid components that have been well established in recent years: are structural lipids, ionizable lipids, pegylated lipids, and cholesterol. So each of these individual components can also be present in a range of different concentrations or ratios, and there are dozens of different examples of each of these different lipid types to work with. So already it's a huge formulation space to explore. In addition to the established kind of status quo particle formulations, you may also want to consider adding things like functional lipids, functionalized lipids, which will add other properties to your particles and potentially influence their targeting within the body. There are also things like process parameters to consider, so the flow rates used in the system, the total flow rates or the flow rate ratio, which will also affect the N to P ratio, so the ratio of your cargo to your ionizable lipids. And then also things like your buffer and dilution fluid used, so what type of buffers use, what pH they're at, and what concentration they're being run at. So this is a huge variable space. When screening, normally once we've found a working process, the process parameters tend to stay fairly locked down, so we can focus really on varying the formulation parameters. But even here, if each of these 9 variables were explored with 10 data points per variable, then theoretically there's a billion unique experiments you could carry out. So it really is an enormous space. Now, there are lots of tools we can use to our advantage to help us kind of narrow down the number of experiments we need to do. We can quickly rule out parameter spaces that are not looking good as we learn from the data sets and the results that we collect. And we can go about planning our experiments more cleverly using things like design of experiments tools so that we can investigate multiple parameters with fewer experiments overall. But either way, this is a really large number of experiments that researchers are faced with. And it's really important that there's an efficient way of churning through all these different formulations without exorbitant cost and time commitment. So that's where Sunscreen comes in. So with Sunscreen, you can load up to 96 different lipid mixes and buffered nucleic acid formulations onto input well plates. Then plan each of your experiments in a convenient spreadsheet before setting your fully automated protocol running, which allows you to walk away and let the system do its thing. So the system uses driving fluids, so ethanol and aqueous buffer typically, to move your precious packets of sample around the system, mix them together at one of our sunnies, meaning you can use as little as 400 microliters for a typical LNP formulation. And using less of your expensive reagents makes a really big difference when you can run 100 experiments a day and hundreds more a week. Sunscreen's sunnies are reusable microfluidic mixers and come in a range of different types. So there's flexibility for different applications and different types of particle formation process. And the flow path is automatically cleaned, which means that we can run completely unique formulations without worry about cross-contamination. The reusability of all of these components as well and the full automation of the systems means we are able to eliminate per experiment consumables. The reproducibility, because of all the automation, is extremely good. And you can see from the data on the slide here that there's less than a 1% CV across 90 repeats here of DOTAP LNPs that we produced across a single run. Those red bars are 6 experiments in which we intentionally left out our pegylated lipid to mimic a formulation that might have poor stability, because when you're screening novel formulations, you have no idea if they're going to work or not. So you don't know if the results will be good or bad. But you can see that if you get a bad result, the recovery of the system is immediate. We don't have any cross-contamination or change in the quality of the subsequent repeat samples. So that means you can screen formulations with confidence, knowing that any variability in your results is actually down to the formulation and not some inherent variation in the process. Now, one of the areas of research where high throughput screening offers extremely high potential for significant advances is in the field of targeted delivery. So for vaccines, localized injection into muscle tissue is adequate for sufficient efficacy. However, the body ultimately processes foreign particles such as LNPs through typically the liver. For cancer therapies or for other therapies that are intended to treat specific tissue types or organs, adjustments can be made to the formulations to direct the particles within the body to improve efficacy. At the Karolinska Institute, the team of Sten Linnerson are working on LNP formulations to target glioblastomas in the brain. And at Unchained Labs, we're always excited to hear about our customer success stories and even more excited to be able to share them. So the Karolinska have been the users of Sunscreen and Sunshine for a number of years. And I'm very pleased to hand over to Razieh Karamzadeh, who is going to tell us more about the work she's been doing.
Thanks, Ben. I'm going to talk about LNP formulations aiming for brain delivery today as part of my postdoc project at Karolinska Institute. In our lab, we've been aiming to know more about brain tumors, and one of the most common occurring malignant primary tumors is glioblastoma, which is representing about 80% of all primary malignant tumors of central nervous system, and the mean age of primary glioblastoma presentation is about 62, and the median survival is approximately 14 months. One of the branches of our studies in the lab is designing lipid nanoparticles delivering nucleic acid into brain tumors, and I've been working to work with DNA-LNPs to target glioblastoma. As we all know, lipid nanoparticles are lipid-like structures which consist of at least 4 different lipids: pegylated lipids, helper lipids, ionizable lipids, and cholesterol. Precise combination of these lipids, in addition to different types and ratio, could result in different formulation. We've been having 2 of the instruments from Unchained lab, Sunshine and Sunscreen, and I've been working with optimizing and establishing lipid nanoparticle production for about 3 years. And today I'm going to show you that we've been making high quality lipid nanoparticles with both Sunshine and Sunscreen, and specifically today I'm going to show some LNP results and formulations from Sunscreen. We've been using different chips and also different settings from Sunscreen. And all of these formulation, I'm going to show some of the LNP characterization parameters like size and size distribution, encapsulation efficiency, and those. And then I'm going to show the functional delivery of these LNPs in vitro and in vivo. Here in this slide, you can see size and size distribution of some of these LNPs that I made in the lab with 2 different chips. In the left-hand side, you can see the LNP formulations from droplet junction chip, and on the right-hand side, you can see the LNP formulations from 5-input chip. And each plot shows LNP size in diameter on the left side, and on the right side is mean polydispersity index. And the first column is one of our reference formulation from Colley's team, which we chose as the control or reference formulations, and the rest are the novel formulation that we are working with. So most of our formulations, we could see that the LNP size is around 100 nanometer in diameter, and the average PDI is about 0.2. On the other hand, I also made some LNP formulations with 5-input chip that you can see in the right-hand side. I could also make high-quality LNPs with size about 100 nanometer in diameter and the average PDI of around 0.2. Here is exactly the same LNP formulations that I showed in previous slide, and in this case, I'm showing you the encapsulation efficiency, which comes from both chips that I've tested. And we can see almost the same and more than 80% encapsulation efficiency in both cases, in droplet junction chip LNPs and also in 5-input chip. I've been also working to make barcoded DNA-LNPs with Sunscreen as well. And you can see here in this slide that I showed some of these barcoded LNP formulations in terms of dynamic light scattering, characterization, meaning size, and polydispersity index. Here in the right-hand side, you can see the plot of LNP size and mean PDI, which shows almost the same amount of, we can see the same size almost, and the average mean PDI is about 0.2. The first column is LNP without barcode, and the rest are LNPs with different barcodes, as you can see. And almost all of them, we got 100% encapsulation efficiency. Next, I would like to also show that these LNPs that we made in our lab works functionally as well, which I've tested some of these in vitro and in vivo in cells and also organates, which I'm going to show. Here is the slide about transfection efficiency of those formulations that I made. And it's ongoing study. So on the left-hand side, you can see some green dots, which are green fluorescent protein positive cells. And it shows the transfection efficiency basically of our immortalized cell. And on the right-hand side, you can see the quantification of this transfection efficiency in 2D culture or this immortalized cell. And it's based on each formulation that I made. So in the upper side, you can see DLS characterization, meaning LNP and PDI. And on the bottom, you can see the corresponding transfection efficiency of each LNP formulations. The other work that I've been doing and testing these LNPs are on the organate on 3D culture, which you can see here. We have been establishing patient-derived organates from patients. You can see in the left part of the slide that we have these tissue from patients and then we established these organates from these tissue. These organates are about 100 to 1,000 micrometer in diameter. I've been also testing some of these LNP formulation that I made with Sunscreen on these organates as well. And we could see some green signals in different organates, and each well shows different LNP formulation that we got. And here is the video live imaging of some of these organates that were transfected with some of these formulations. And on the video on the left, you can see basically the transfection efficiency overnight with one of our LNPs. And we can see almost increasing signal during the night. And on the right part of the video, the other LNP that shows the dividing cells during the imaging. And I've been working also to quantifying these GFP signals in 3D culture in these organates. And we were successfully got transfection efficiency and quantification of these LNPs. One of the other things that we also did was testing these LNPs in vivo as well. So what we've done was injecting these some of these LNPs intravenously and via the tail. And here in this slide, you can see the IVIS results showing the biodistribution of lipid nanoparticles in a whole body. We have control that we injected PBS, and we have other mouse that we inject di-LNP. And we could see different biodistribution based on different LNP formulation. And on the right-hand side, you can see some of the organs that have this signal and showing these LNPs. And you can see also liver and also brain and other organs that could successfully got these LNPs. And you can also see that these LNPs that we made with in vivo also had the same characterization almost in terms of size and polydispersity when we concentrated in and injected into mice, at least with dynamic light scattering characterization. Here is another example that we've been working on, and here we injected luciferase LNP, LNP that contains luciferase gene. And we injected around 5 microgram intrathecally through the spinal cord. And we could see the expression of luciferase gene in spinal cord and dorsal root ganglions. And you can see the signal here in the spinal cord. And these formulations also, in particular, this one that I'm showing, if we want to see the dynamic light scattering characteristics, the LNP size were about 100 nanometer and the mean PDI was around 0.1. And the encapsulation efficiency in this case was also 100%, which we successfully could got these results. These are ongoing studies, so we've been working on. And yeah, we got the very high quality LNPs, and we are satisfied with the results as well. With that, I would like to thank Stanley Narson Lab at Mol Neuro, Department of Medical Biochemistry and Biophysics, and also Marina and Doris, who did the in vivo experiments. Thank you for your attention. I'm happy to take any questions.
Thank you so much, Razieh and Ben, for a great presentation. We're now going to start our Q&A, so I would like to invite you to join me on camera. Our first question is for Ben. It is, what stage do you switch from using Sunscreen to Sunshine?
So that kind of depends on what your workflow looks like. So one example might be volume. So with Sunscreen, obviously we're focusing on being efficient with material and trying not to use too much of your kind of precious reagents, or at least not more than you need to. But at some point, you're going to want to produce larger volume samples. And with Sunscreen, your upper limit is a 2 milliliter deep well plate. So if you want to collect more volume than that, then Sunshine will allow you to do that both in a kind of similar automated protocol mode, but then also just switch to continuous mode to produce liters of product if you need that much volume. So the other thing might just be, if your lab has kind of parallel workflows, one for kind of the high throughput screening of lots of different formulations, and then separately a team of people kind of honing and tweaking the really promising ones, then you might not want to be dedicating your high throughput screening instrument to doing lots of kind of very similar repeats of a similar formulation, and actually migrating that work onto the Sunshine with a view towards transferring to larger volumes might be more efficient use of the instrument. And that way the Sunscreen can be set back to work, churning through samples, hunting for the new next winning formulation.
Thank you so much, Ben. Our next question is for Razieh. Are there other brain-related disease types these formulations could be used for?
Yeah, good question. Yes, we can also adjust this LNP formulation to target other organs as well. Yeah, it's possible.
Great, thank you so much. Our next question is for either one of you. It is, when screening different LNPs, what is the most important parameter? Use different ionizable lipids or vary the ratio between the lipids?
Yep, I can take that. Both of them actually are important. Depends on the target and also the formulation. Yes, both can be adjustable. Yes.
Thank you so much for taking that one. The next question is also up for grabs. It is, how is the translation from Sunscreen HD formulation to scale-up formulations? Are there any differences on the LNP feature or transfection efficiency?
Yeah, maybe I can answer this one as well. So I've tested particularly some of my LNP formulations in a small and large scale in terms of transfection efficiency. So I couldn't see any significant differences. So that should be okay, I think.
And that agrees with the testing that we've done in-house as well, Razieh, of just comparing similar formulations and scaling them up through the platforms. We get really nice transferability.
Thank you so much to both of you. Our next question is, what is a typical NP ratio or MM ratio of these DNA-LNPs?
Yeah, so these are formulated with different ratio, but particularly those that I showed in those slides were 6, NP ratio 6. Yeah.
Thank you. Our next audience member has a few questions. They're asking, do you perform online dilution or high throughput dialysis or anything else to reduce or remove ethanol before application in vitro, in vivo? So does someone want to answer that one first, and then I can move on to the next one?
This one, I think that I could just generally answer that. So about it depends on DNA and RNA-LNPs. In my case, I would prefer dialysis to change the buffer. And also the other thing that would be also nice to consider is a dilution chip that could help a lot to dilute the ethanol from the LNPs. So both dialysis, I would say, and also using a dilution chip would be nice to try.
Right. Their next question is, do you know anything about stability of the DNA-LNPs themselves during storage?
Yeah, good question. This is a very challenging area, I would say. There are some publications about this, but I haven't tested actually storage yet for these DNA-LNPs. But yeah, I would recommend to check different publications for this.
Thank you so much, Razieh. Our next question is, how much DNA was injected via IT?
Yeah, it's 5 microliter in volume, and I could also check because we tried different concentration. So it was 5 microliter, and yeah, let me quickly check. I can just a minute. Yeah, 5 microgram. Yeah.
Thanks so much for checking that. This next question is there any difference in the LNPs used for DNA and RNA?
Yeah, I would say this is also a challenging question. So what I've tried was DNA, but I think that these formulations that I've been working on, it is also possible to use them for RNA LNPs with a little bit of adjustment. So it couldn't be any problem, I would say. But I haven't tried all of them.
Thank you so much. I think this next question is also for you, Razieh. It is, did you also test other chips instead of the Sunny XT or the 5-input chip? What led you to choose these 2 chip geometries? And did you observe a bad behavior for the DNA-LNPs with other chips regarding size/PTI?
Yeah, I mean, I quickly checked most of the chips because I was curious. I would say I couldn't see any bad effects, but one of the things that I can basically recommend is to check every parameters and adjust it accordingly. So not all the chips works with a specific ratio. So would be good to check that one as well. And no, I couldn't see any bad effects. And why I chose those, because I was much curious about the differences between the chips and the results of LNPs, basically. And yeah, it was nice.
Great, thank you. One last question before we wrap up. This question is for Ben. How do you change between mixer styles? Do I need to take apart all the system tubing?
No, so the chips changed recently. So the new Sunnies have a consistent connection. So it's quicker now to switch between the types. So as Razieh was just saying, if you are kind of just starting with them and trying to work out, I don't know, which one's going to be the best for my formulation, it's something that's now quick to try, just to switch between these different geometries and run very similar experiments. And generally speaking, that process is an initial check to work out, with the typical sorts of formulations you're working with, what kind of mixer will get you in the right kind of size range and flow rate range for the work you want to do. And then, as I said at the beginning, you tend to kind of fix that down for a period while you then screen a lot of different formulations. And then when you move on to, say, the Sunshine instrument, when you're looking to kind of optimize your the formulation you identified through throughput screening, you can then revisit those different mixer types and see if there's a different one you could maybe use to eke out slightly improved performance or different results from your efficacy studies.
Excellent. Well, thank you so much, Razieh and Ben, for answering those questions. Unfortunately, that is all we have time for today. Any questions we didn't get to, we will reply to you by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the Resources tab on the right-hand side of the screen or in the on-demand email. All that's left is to thank Razieh and Ben once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.